Hello everyone, this is your colleague Raghad, and today I'll be talking about asthma. It's a lecture in immunology. So before we start, what's asthma? Asthma is a chronic inflammation disease. So is it reversible or irreversible? It is reversible, but to know that COPD is irreversible. Asthma occurs due to allergy with excessive harmful response. That should not happen, and this response should not be there. According to the classifications, asthma is atopic and non-atopic types. It has atopic and non-atopic types. The atopic types, it has something that triggers it from outside, but the non-atopic one, it's from inside the body. The atopic ones, it's mostly genetic. It has elevated IgE, but the non-atopic one, it's mostly environmental, such as infection, stress, cold, exercise, chemicals, and it does not have elevated IgE. Moving forward to how can we differentiate between them? By skin prick test. So we have a test in the lab. We can differentiate between the atopic and the non-atopic types of asthma. It's called skin prick test. The results, it should be negative. We have negative and positive. The negative is non, non-atopic, but the positive one is atopic. Now let's dive deep into the atopic type. Before we start, we have something called wheel and flare reaction. This reaction is here in this picture. We have this test that we were talking about, which is prick test. Allergens induced into the skin by a needle. For example, this is the skin and this is the needle. They break the skin by this needle. Then the previously bound IgE on the mast cell get activated. Then this mast cells released mediators, and blood vessels causes local swelling, which is the wheel, this one. And then dilation of, or plus we can say, dilation of blood vessels on the edge of swelling gave flare appearance, which is this one, the dilation here. So this reaction depends on the IgE and mast cells. Moving forward to the immunity in the asthma, we have adaptive and innate. The adaptive immunity, which is B and T cells. Basically here we have B cells, CD4, which will differentiate into T helper 2. And innate immunity, which is especially the mast cell, neutrophils, and the eosinophils. Ready to dive more? Moving to the first encounter in the asthma, we have multiple encounters. So the first time this antigen, I don't know what is this, it enters the body, there's no reaction, production of allergen-specific IgE happens here. T helper 2 cells will release interleukin 4, 5, and 13. Innate immunity will produce interleukin 2 also. Then this IgE binds to the Fc receptor on the mast cell, which is this one, on the mast cell, making it ready for the next exposure. Yani it's there, the gun is there, but it did not shoot. And there's also, it's responsive to bronchodilators. Yani when I give them bronchodilators, they'll be okay. The patient will be okay. Then moving to this antigen, it came again. I know what is this now because I have already the IgE in my body. The second encounter and onwards, we have the allergen that dives into the mucosa, in the submucosal mast cells, in lower airways. And this activation results in mediators release, which will cause calling eosinophils and inflammations. Yani this antigen came, entered the body in the lower airways. This antigen again will cause eosinophil and inflammation cells to come to the place where this antigen is present, and bronchoconstriction. And this one is, it responds to steroids. Moving forward to the immediate phase and the late phase. So we have two phases, the immediate and the late phase. Immediate phase, it has bronchoconstriction and it's type 1 hypersensitivity because we have allergen and IgE. But in the late phase, T helper 2 eosinophils came due to interleukin 4, 5, and 13. To specify, interleukin 13, it drive mucus cell metaplasia, but interleukin 4 and 5, they are more to macrophages. So what are the mediators that are released by mast cells? We said here that mast cells produces some mediators. So what are these mediators? We have histamine, heparin, proteases. Proteases is an enzyme, so it causes remodeling. Heparin inducing bradykinin. Then the histamine, we know that it increases the permeability of the blood vessel. We have also cytokines, chemokines, and lipid mediators. Cytokines such as interleukin 13, interleukin 5, and interleukin 4. To mention that interleukin 5 is special for the eosinophils. We have also the chemokines such as CCL3, lipid mediators, which are C4, D4, and E4. Moreover, these things, we have also prostaglandin D2, interleukin 6, interleukin 8, and TNF alpha. Moving forwards to the cytokines, let's dive into the cytokines in asthma. It's produced by T helper 2, which needs activation, so it's adaptive. We have interleukin 4, 5, and 13. These are the main ones. Interleukin 4, it's for production of IgE and activates mast cells and eosinophils. Interleukin 5, it's specially for eosinophils, so it activates, attracts, form and persists the eosinophils. We have also the interleukin 13, which induces inflammation, stimulates mucus hypersecretion, and induces subendothelial fibrosis and remodeling. So this one is responsible mainly for remodeling. Moving forward to the eosinophils in asthma, we have also production is inhibited by interleukin 10, the brakes, if you remember. We have also something special in asthma made by eosinophils, which is the charcot-Leyden crystals formed from eosinophils granules. Cause tissue damage, of course. Cause tissue damage. Eosinophils produce lipid mediators, cytokines, enzymes, ROS, granule-derived proteins. So, again, eosinophils, it's stopped by interleukin 10. It produces charcot-Leyden crystals. It causes tissue damage, and they produce lipid mediators, cytokines, enzyme, ROS, granule-derived proteins. For the remodeling part, it's mediated mainly by TGF-beta, and in atopic asthma, we have the T helper 2, which will produce interleukin 13, as mentioned here. So this will cause remodeling in atopic, which is interleukin 13 produced by Th2. We have the non-atopic asthma. Meanwhile, in the non-atopic asthma, we have Th17. So this one will cause remodeling. Again, in remodeling, it's mainly by TGF-beta. But in atopic asthma, T helper 2 produce interleukin 13. And in non-atopic one, T helper 17, which will cause remodeling. What is this remodeling? We have angiogenesis, mucus plug, goblet cell hyperplasia, subendothelial fibrosis. This is the remodeled vessel. Why asthma develops? Due to disturbance in balance between the T helper 1 and T helper 2. T helper 1 comes from the environmental exposure, but T helper 2, the baby can get it from the mother's body. So, moreover that, it is pro-allergic. From here, we have the hygiene hypothesis. The hygiene hypothesis states that increased hygiene, vaccination, and use of antibiotics may alter the human immune system, so it will react in a wrong way to harmless substances. Let's see this picture. In normal situation, T helper 2 comes from the mother to the baby, and the T helper 1 comes from the environmental exposure, so it increases by experiencing something from outside. It's full or it's equal at the about year of 2, yani 2 years old. But in the delayed maturation of T helper 1 capacity, The T helper 2 is there from the mother, but there is no T helper 1 because there is extra hygiene on the baby. So Treg cells also, we know that these are the brakes of the immune system which will produce interleukin 10 and TGF beta. We said that TGF beta is there in remodeling. So how it's produced by T regulatory cells? The answer here is from ChatGPT. While T regulatory cells secrete TGF beta primarily, the immune suppression will resolve this inflammation. The same molecule seamlessly acts on structural cells like fibroblast and smooth muscles in asthmatic airway to drive excessive collagen deposition and proliferation, causing airway remodeling and fibrosis. And that's it for today. Good luck.